Hi dear students, welcome back to one more session on immunology, and let's continue with the second session on antigens. In the previous session we had mentioned about what is an antigen and how is it different from an immunogen also. As we use the term antigen, it is commonly referred to as a self and a non-self substance which could elicit an immune response and react with the products of that particular response. That is a common term that we used, or that is a way how we define a antigen. But specifically talking, the term immunogen is a one which is more suitable when a substance is capable of eliciting an immune response as well as reacting with the products of that response. Though we use the term frequently antigen, the term immunogen is a one which is more suitable for this case. Now, what do you mean by immunogenicity? Immunogenicity is the ability to induce immune response, and it could be either be humoral or the cell mediated immune response. Now there do exist what is the difference between antigenicity and immunogenicity. Antigenicity is the ability to combine specifically with the final products of the above responses, that is antibodies or the cell surface receptors and all that. So if you're going to strictly define antigenicity and immunogenicity, you can divide it into two terms. If both immune response detection as well as combining with the products of the immune response is being carried out, that means it is immunogenicity. Okay, so only if they can combine with the final products of the response, it is referred to as antigenicity. So you have to put a demarcation that if a substance is being immunogenic, it will what? It will elicit an immune response. That is, it will either trigger the B cell activation and induce a humoral immunity, or it will activate the cell mediated immune response, as well as it would also interact with the products of these immune responses. That is, either it will interact with the what? Antibodies, or it will interact with the what? The activated effector T cells. So a substance is immunogenic indicates that it will elicit an immune response and interact with the products of that particular response. But the term antigenicity is only meaning that it has an ability to combine specifically with the final products of the immune response. And so when you think about this concept, you can see that although all molecules that have the property of immunogenicity also have the property of antigenicity, but the reverse is not true. So if you say that a substance is antigenic, it doesn't mean that it is immunogenic also. That is, when it is antigenic, it can only interact with the specific the products of the immune response, but it does not trigger a what? A immune response. And you can see that there are small molecules called the haptens, which are antigenic, but incapable by themselves of inducing a specific immune response because they are being found to be, because they do not have the property of immunogenicity. So do understand what is the difference between antigenicity and immunogenicity based upon the whether they are capable of triggering the immune response or not. Now, if you go to consider the different types, so keeping this concept in your mind, we will just go to talk about the different types of antigens. We do come across different types of antigens within our body, and let's move on to what are they. We have haptens, reactive antigens, Phosm antigens, cluster of differentiation antigens, T-dependent antigens, T-independent antigens, and let's explore what are they. Coming to the first one is a hapten. Many small organic molecules are not immunogenic by themselves, but sometimes they become immunogenic if they get bound to large carrier molecules such as a protein. And these small antigens are called the haptens. That is, Hapten means to bind. Okay, and this was developed by Karl Landsteiner. And what happens is they serve as incomplete antigens, and only when they combine with a carrier molecule or what do you say? Only when they bind with something else, they become highly immunogenic or they become immunogenic. In other cases, they will be what? They would be serve as an incomplete antigen. Now, for example, we have your nitrophenol, which is a hapten. When it combines with serum albumin, it becomes a complete antigen so that it can induce a immune response. And some acts as haptens in some animals, whereas act as complete antigens in the other. Now, for example, if you take pneumococcal polysaccharide, it is a complete antigen in animals such as mouse and man, but it is a hapten in the case of rabbit. So, when you consider a hapten, it might serve as a hapten in some animals, but at the same time, it might serve as a complete antigen in other animals also. Another example is penicillin. By itself, penicillin is a small molecule and it has not been found to be antigenic, but it becomes when it combines with the serum proteins of a sensitized individuals, the resulting molecules become immunogenic and it activates the lymphocytes and initiates a severe and sometimes a fatal allergic response. Now, the second class of antigens which we come across is the cross-reactive antigens. You should remember that cross reactions can occur between antigens which bear stereochemical similarities. Cross-reacting antigen is one which is capable of binding to the antibody produced in response to another antigen. Now, for example, you have a heterophile antigen that is which exist on the surface of unrelated plants, animals, and bacteria, but are identical so that the antibodies to one will cross-react with the other. Now, human blood A antigen is cross-reactive with the antibodies of pneumococcal polysaccharide type 14. And the human blood group antigen B antigen can be cross-reactive with antibodies to certain strains of E. coli. So a cross-reactive antigen is an antigen which has been found to be similar in different. They have stereochemical similarities between what different type of organisms, and so what happens is the antibodies produced against one antigen could be able to interact with the other antigen also. Now here, if you have an antibody which has been produced against human blood A antigen, okay, on the surface of RBC there is a antigen called the A antigen. Okay, the antibodies which are produced against that A antigen can also interact with a pneumococcal polysaccharide type 14. Pneumococcus is a bacteria and there is a polysaccharide in that. And so what happens? The antibodies produced against the human blood A antigen and against the pneumococcal polysaccharide 14 type 14 is also been found to be similar. So that's what a cross-reacting antigen. It is mainly because the antibody is capable of binding to two similar antigens. Now let's move on. And the next one which we have is a Phosman antigen. It was found by John Phosman, and these are glycolipid antigens originally described to be present on the most tissues of guinea pigs but not on their RBCs. And this antigen is being found to be truly heterogenic as it is found in the RBCs of many species of horse, sheep, dog, whale, cat, toad, streptococcus pneumonia, etc. And it has been found to be a hapten. And what does it means? On the surface of most of the tissues of the guinea pigs, you can see this antigen, but it is not. And same antigen is also being found to be what in the RBC of horse, sheep, dog, cat, whale, and all, and serves as a hapten And you can see that although humans are considered to be as Phosm negative, it is found that the gastrointestinal mucosa of some people do contain the Phosm antigen. So, why it has been explained over here? Because it is of significance, which was uh, and it has been found to be commonly found in various type of organisms also. Then another antigen is the CD, or the cluster of differentiation, which we always come across in different cells. Now, the lymphocytes and other immune cells bear cell surface proteins which have a specific role in the intercellular communication. And these are called the cluster of differentiation molecules or the cluster of differentiation antigens. The CDs are the cell surface proteins, and many are receptors, and they do have biological as well as diagnostic significance. And you can see that in the case of HIV, they have a CD4 molecule as a receptor, and if a cell is being considered to be as a stem cell, it will have a CD34 receptor. CD34 would be present on its surface, and CD4 MHC class 2 co-receptor is being found on the T cell, and CD8 is being found in the what? On the cytotoxic T cell. So like that, to differentiate the different type of biological as well as uh, biological cells, you can use a cluster of differentiation antigens. So to differentiate between the leukocyte subpopulations, we mainly use the cluster of differentiation antigens. These are some of the antigens. Now the T cytotoxic or the CD antigens of the T lymphocytes. If it's a T helper cell, it will have CD4. You know that. And if it is a TC cell or a cytotoxic cell, it will have a CD8 molecule on it. And if you go to consider the B cells, they also do have CD molecules, but it is not the CD4 or the CD8. So it is helping to differentiate between the lymphocyte subpopulations. We can also classify the antigens based upon the as T-independent antigens and the T-dependent antigens. Now, sometimes a few antigens can trigger the B cells to produce antibodies without the T cell cooperation. These antigens are called the T-independent antigens or the T-independent and this stimulation is referred to as a T-independent antigen triggering. Now, for example, bacterial lipopolysaccharides as well as certain tumor promoting agents, what happens? The antibodies are being produced against this even without the help of the T lymphocytes. So, the T-independent antigens are those which do not require the help of the T lymphocytes to trigger an immune response. And the T-independent antigens, usually they tend to be polymeric, that is, they are composed of repeating polysaccharides or protein subunits, and the resulting antibody generally has a low affinity for the antigen. Now, we do have another class of antigens or some antigens which requires the specific action of or the help of the specific action or the help of the T cells to trigger the B cells. Such antigens are referred to as a T-dependent antigens. So, when when we go to consider a antigen, and if it cannot develop into the plasma cell, sometimes what happens, it will not be able to trigger a B cell to produce the antibody producing plasma cell without the help of a T helper cell. So when you need the help of a T helper cell for the B cell to be triggered, that is referred to as a B dependent antigens. And they could include particles such as bacteria, foreign RBC, certain proteins, and Hapten carrier combinations do come under the category of the D dependent antigens. Then I would also like to say about what is affinity and avidity. Avidity, sorry. Antibody affinity, it relates to the strength with which an antibody binds to its antigen at a given antigen binding site, whereas avidity of an antibody, it relates to its overall ability to bind to the antigen to all the antigen binding sites of a particular antigen or of a of a antigen. Yeah, that's it. So the strength of the antigen antibody interaction is mainly being explained on the basis of this affinity and avidity. If you mean what is it now, avidity means the overall ability to bind to all the antigen binding sites, whereas antibody affinity, it refers to the strength with which the antibody binds to the what antigen at a given antigen binding site. So I guess you got an idea about what is an antigen. The term antigen is, though it is commonly used as immunogenic, as well as capable of interacting with the products of the same. It is more suitable. This definition is more suitable for the term immunogen rather than an antigen. An antigen is, if you go to talk precisely, antigen is substance which could interact with the products of the immune response. But the term immunogenous is the one which is more suitable when it is capable of triggering an immune response and capable of interacting with the products of the immune response. And we have an overall idea about the type of antigens which we come across within our immune system. With this, we come to an end of this session. Thank you for now.